Okay, so today, the limbic system. Now, this week we're going to do the limbic system and just try to get an idea of the main connection. So I only have one board today, so lots of parts. I don't expect you to remember all the parts. I can't necessarily remember them all exactly myself, so I won't expect you to. But some of the main parts, like the hippocampus, that we're going to talk about in more detail, and we'll also talk about head direction cells and part of the hypothalamus and other places. Want to remember those. So why is it called the limbic system? So if you go around sort of on the medial wall of the brain, so there's the corpus callosum like that, and there's the cingulate sulcus is sort of sitting right there. But if you look at the medial wall of the brain, the limbic system, limbus is a skirt, and so this whole area around the corpus callosum, and you could then going down into the hippocampus and something like that. That's supposed to be a skirt. So, like I said, people who do neuroanatomy, they spend maybe too much time looking through the microscope, and so they thought that looked like a skirt. So the limbic system there's a bunch of structures we're going to talk about that have, there's a lot of connections sort of going around like this in a loop. And the reason for that is because of the formation of the temporal lobe. So those connections used to be kind of straight, but then as the temporal lobe sort of curls around like this, that's why you get that. This is medial view. And so there's a whole bunch of structures we'll talk. There's some structures that are sort of back here that are sort of doing sort of visual stuff, and there's orbitofrontal cortex right here. There's the hippocampus, the amygdala. So we'll talk about a lot of those structures. So that's the limbic system. So is this like a really completely separated system? And the answer is, of course not, because it has connections to the red nucleus. It has connections to sensory cortical areas. It has connections to motor cortex. So it's kind of it's just a convenience of grouping some structures together. They aren't like cut off from the other structures. And in particular, what we'll see is that they need to actually be connected to those other structures, because that's kind of the whole point of the limbic system. And so what is the whole point of the limbic system, stepping out to the most sort of macro level? And so the whole point of the limbic system is to monitor your internal state. So your internal state's quite complicated. It's got you think of things like thirst and hunger as a real sort of basic part of the internal state, that every animal has to sort of get enough food and keep the engines running. But it turns out over the past 20, 30 years, people have found that a huge chunk of the limbic system has to do with basically what you might call an inertial guidance system. So the limbic system represents not only how hungry are you, how thirsty are you, how angry are you, how interested in sex you are, how interested in all these different sort of appetitive things, but it also has a huge chunk that is actually related to where you are in the environment, exactly where you are. What direction is your head pointing in the environment? And that turns out to take up a huge chunk of the limbic system, which makes sense that that's going to be very closely related to things like where the food is, and if it's a dangerous situation, where's a good place to go? And so on Wednesday and Friday, we'll talk about the work on place cells and head direction cells and grid cells and all these different parts of the limbic system that seem to involve telling you, representing your internal state of where you are in the environment. So that's just as much of an internal state as how hungry you are is an internal state. But if there's no connection between I detected I'm hungry and the rest of the brain that knows how to sort of find food, obviously the limbic system, the limbic system can't just be an island. It has to sort of talk to the whole rest of the rest of the brain. So that's what we're going to show. How does it connect up to the rest of the brain? So we'll have like a big diagram about like this big, and then a smaller one down here. So let's start off with the. So I'm going to organize it by forebrain. So forebrain, thalamus, and midbrain and below. So starting off, a classic limbic system structure is the septum. So the septum, it's a small structure in humans, but it's very important. If you lose it, lots of things get broken. But another part is the basal forebrain. Now, you remember like 2 lectures ago, we had a nightmare diagram of the connections of the striatum. So just pack all those right into there. So that's the basal forebrain, all those connections that radiated out to the superior colliculus and the thalamus and motor cortex. That's there. And then there's another big chunk of structures that are sort of classic limbic system, and that's the hypothalamus. So the hypothalamus has, so there's, let's put the number, so there's only 2 structures in the septum. So that's good. There's more here. We'll say 25, okay? Or maybe that's a little bit too many. We'll say 15. So the hypothalamus has a whole bunch of little structures in it as well. There's at least 20 little substructures in the hypothalamus. Some of them are involved in monitoring day-night cycles, monitoring fluid balance inside your body, how thirsty are you, monitoring, hunger. And, these systems are incredibly well tuned. Like if you look at animals out in the environment, all animals out in the environment have the right weight. All lizards maintain their body weight correctly. All cats out in the wild maintain their body weight correctly. Bears. Humans screw up because we're in this weird food environment that has these hyper attractive foods that kind of are designed to sort of get around all our limbic system things that are designed to keep you right on the money. Animals eat the proper number of calories to within 1% on average. That's how accurate they are. So all you got to do is just eat the right food, and then everything, just let your limbic system do its thing, and it'll sort of take care of business. So that's the hypothalamus. And then there's a group of structures down here called, we'll just call it the limbic midbrain. And we'll talk about one. Midbrain. So we'll say there's 6 structures in the limbic midbrain, but we'll talk about one in particular, the pedunculopontine nucleus, PPN, because that's a big important one. So if you look at these structures, you see there's a giant highway of fibers that goes back and forth between the basal forebrain and the septum, the hypothalamus, and the limbic midbrain. And this thing is called the medial forebrain bundle. Medial forebrain bundle. Medial forebrain bundle. And it's a highway. It's got like 50 things going every which way in it. And when you look at the brain, one of the things, you often see these giant highways like this, like the medial forebrain bundle, and stuff will come in, come out here, go in, come out here. It's probably has something to do with the fact that when axons are actually going someplace, they kind of like to go and hang out with the other guys that are all going the same direction. And you have a tendency to see relatively right angle crossings between axons. Maybe the reason for that is that, like, if this guy's growing along, if he's at a right angle, he has a better chance of getting through instead of going like, if you come in like this, you're trying to go here, but instead you might go, oh, I'm going to go with these guys. So when you look at the texture of the brain, you often see these sort of almost right angle crossings between bundles of cells. You see the same thing in the white matter. You can see it down in the brainstem. And so this is an example of one of those giant highways. So, the medial forebrain bundle, and it's also down here. So that's the original pleasure center. So is it the pleasure center? Absolutely not. So what was this? Where did this come from? This came from those experiments where you put an electrode in the medial forebrain bundle, and like I said, there's 60 things going every which way. So it's not specific at all. So if you put an electrode in there and give a rat a little shock, what the rat will do is stop everything and just say, whoa, what was that? If you hook up that shock to a lever, the rat will just stop eating, stop drinking, stop everything, just press the lever to get shocks. And so this was interpreted as the pleasure center, but it's more like sort of the craving for cocaine center. It's like, you gotta do it. It's not necessarily pleasurable. It's this incredible motivating stimulus that says, like, do it again, or do whatever you did there. That's good. Just keep doing that. So it's a very, it's the motivational system of the brain. Now it has several different outputs, and one of the major outputs that comes out of the hypothalamus, you can sort of think of it as a motor output, is the pituitary. And so the pituitary, so if you look at the pituitary, so that's called the hypophysis or hypophysis. Hypo, the underneath physis. And so that has two parts. So there's the posterior part, and the posterior part is actually, it's called the neurohypophysis, and that actually has that actually has these giant neurons in it that are so big that they can actually release enough hormone-like substance into the bloodstream to affect things, to affect the body, just single neurons. And they release oxytocin and vasopressin, which is something that's involved in blood pressure and fluid regulation. But most of the parts of the pituitary, most of the hormones related by the pituitary are the anterior pituitary, and that's called the adenohypophysis. Adenohypophysis. And that guy, that one has neurons in it, specialized neurons that dump hormones into the blood. So the neurons come down, hit these specialized sort of hormone dumping cells, and then the hormone dumping cells dump stuff into the blood. That's where most of the hormones come out of. There's only two coming out of this first one. So So you can think of that as it's kind of a motor-like structure because it actually, makes the body do things like, get really excited or turn up your heart rate. Now, these down here, the limbic midbrain, we'll see these guys can actually access skeletal muscle so that they can actually move the body around like a motor cortex directly without necessarily having to go to motor cortex. So we'll talk about that when we talk about the pedunculopontine nucleus. So if you want to read about the medial forebrain bundle, the guy who wrote that book, Rudolf Nieuwenhuis, that Dutch guy who wrote that neuroanatomy book, he's the dean of the medial forebrain bundle. He's studied all the different pathways going every which way in the medial forebrain bundle. Okay, so that's the medial forebrain bundle, septum, basal forebrain, hypothalamus, and limbic midbrain structures. Okay, so now let's move up to another structure in the forebrain, which is the amygdala. Let me just put that down a tiny bit. Give me a little more space. So here's the amygdala. And then there's the hippocampus. So here's the hippocampus. So the amygdala is supposed to be shaped like an almond. Amygdala, that's the almond-shaped nucleus. It doesn't look particularly like an almond shape. And then the hippocampus is the seahorse-shaped structure. So there's a lot of substructures in the hippocampus. We'll say, if you sort of think of the greater hippocampus, there's about 14 separate fields in there. And the amygdala is also subdivided. There's 3 main subdivisions of the amygdala, but within them there's multiple subdivisions. We'll say 10 distinct subparts of the amygdala. And so one of those big pathways that sort of circles around on the midline, sort of near the midline, is called the fornix. And the fornix, it projects from the hippocampus down to the hypothalamus and especially to the septum, but also to the basal forebrain. And it's a highway, and so stuff's coming back the other way also like that. So there's another pathway that's going that way. So so that one is called, this guy is called the fornix. And there's another pathway that's running virtually just right on top of it called the stria terminalis. And that's a connection between the amygdala and these guys. So the amygdala goes, and there's a projection coming back to it. The amygdala goes to the hypothalamus and back. And that guy's running pretty much in the same, they're just on top of each other, but that one's called the stria terminalis. Stria terminalis. It's so different than fornix, yeah. And so this, and these almost make a 360 degree loop. So they start in the fornix starts in the, if we look at, something like this, here's so the fornix starts in the hippocampus. Here's the hippocampus. It starts here, and then it sort of like comes up like that, and then it goes almost all the way around until it hits the septum. So it's almost a 360 degree loop. You might wonder, like, why don't you just take the shortcut? And it turns out that's not lost on the brain. There's a few connections that come out of the amygdala that just take the shortcut. They say, like, why do we have to go all the way around? Let's just like punch right through. So those are the two, so the fornix and stria terminalis. And then we got some other parts of the of the thalamus. So there's the epithalamus, or the habenula. So that's a relatively, so remember that was, the hypothalamus was the bottom quarter of the thalamus, then there was the ventral thalamus, that would be like the subthalamic nucleus, and then there was the dorsal thalamus, that would be like 50 things, like the lateral geniculate and a whole bunch of other stuff. Medial geniculate, ventrobasal nucleus. And then there's the epithalamus. So this is and that's the top quarter, although it's much smaller in the in the adult than the dorsal thalamus. There's only two parts of that guy, and that one is hooked up to the pineal gland. So basically this guy is hooked up to the pineal, which is also called the epiphysis. Epis, on top. Epiphysis. So the pineal gland, if you look at something like a lizard, you go out, if you find a dead lizard out in the garden, and it's not too stinky, just pick it up and you'll look on the top of their head, there's actually a little eye. So they have an eye on the top of their head that's directly hooked up to the pineal. And the purpose of this is, it's not a very good eye, it's just like, is it daytime or nighttime? And it turns out that in most animals, like say a rat, the pineal is still light sensitive because light will get through the skull of a thin skull of a rat. Humans, unfortunately, the skull is so thick that the third eye has had to be discontinued. It's no longer light sensitive because it's completely dark inside the human head. But this system is still heavily involved in day-night cycling. And you can see big bundles running up and down from this structure, and so there's a big bundle. You can see it in a turtle. You can see it in a human. It's a big bundle of fibers that hooks up the epithalamus to the limbic midbrain, and that one is called the habenulopeduncular tract. Habenulo, because it's one of the things that's going to I spelled it peduncular tract. One of the things it goes to is the pedunculopontine nucleus. So that's this guy. And then there's another big bundle that's going up to the septum from this. So that one is called the stria medullaris. It's running pretty much along with all these other guys up here. Stria medullaris. So, and like I said, there's still very visible bundles in a human. So it's not just just in rats. And so there's not too many more parts. So the dorsal thalamus is also involved. So generally, if you look at the thalamus, it's kind of mapped out more or less the same as the cortex. So the back parts of the thalamus go to the back part of the cortex. The middle parts of the thalamus kind of go to the somatosensory cortex, and the front part goes into these limbic structures of the cortex, like orbital frontal cortex and the hippocampus. So this guy is the dorsal thalamus, so equivalent to lateral geniculate. So the dorsal thalamus, and how many parts do we have there? Maybe like 6 nuclei at least, like the ventral anterior nucleus and the mediodorsal nucleus. We'll talk about that one when we talk about head direction cells. And that has a big bundle. You can see it in a turtle or in a human that is going and connecting up to the hypothalamus. And so what is that one called? That one is called the mammillothalamic tract. And so why is it the mammillothalamic tract? It's supposed, if you look at the mammillary bodies, those are sort of breast-like bodies, on the bottom of the hypothalamus, this is a big tract that goes between the dorsal thalamus and those hypothalamic things. Now, back in the day when I was studying neuroanatomy, we didn't really know what the mammillary bodies did. I just assumed it was just more drug, sex, and rock and roll, and thirst or something like that. But no, it turns out the mammillary bodies, which are a big chunk of the hypothalamus, are actually heavily involved in this head direction system, and so they're representing to the animal essentially what compass direction relative to, it's not actual compass direction, but it's like compass direction relative to features in the environment. What direction is its head pointing? So that the cell will respond just when my head is pointing this way, but not when my head is pointing that way in this particular environment. And so that's another pathway there. Now, the dorsal thalamus has big projections also to the hippocampus. So the dorsal thalamus doesn't only go to the neocortex; it goes to the it also goes to the hippocampus, and there are projections to the amygdala also. Big projections back and forth to the amygdala. One of these ones I said was the one that takes the shortcut, where there's one called you have to remember this, the ventral amygdalopetal pathway. So it's the ventral pathway that's fleeing from the amygdala, and that's the one that just punches right through it. Says like, screw this, we're just going to go straight. So now the hippocampus is also heavily hooked up to higher level cortex, and also the so let's make a nice big tertiary cortex. We'll call it sort of higher level cortex tertiary. That means like sort of third level tertiary cortex. And there's a lot of structures in higher level cortex. We'll say like 30 structures here. But there's a big projection that comes into the hippocampus from higher level cortex. And there's also an equally big projection that goes back. And similarly, there's a big projection that comes from higher level cortex into the amygdala, and a projection that comes back. So if you look at something like a rat, it's even primary cortex that goes into the hippocampus. So V1, primary visual cortex in a rat, goes straight to entorhinal cortex, which is part of the hippocampus, sort of one-stop shopping. It just gets right there. It's not a big projection, but there's definitely a projection from primary visual cortex. If you look at a monkey is a little more hierarchical, has more cortical areas, especially visual ones, and primary cortex doesn't go to the hippocampus. But there's a weak connection from primary cortex all the way to the amygdala, even in a monkey. So, but most of the connections to the amygdala and the hippocampus in a primate or a human are coming from third or fourth level areas in the cortex. Okay, and so and then of course there's a giant projection from the dorsal thalamus, up to, that's the main one we were talking about, and that one is called the LFB, the lateral forebrain bundle. And so if you look at something like a turtle, you can sort of see these two bundles right next to each other. In a human, it's a little more complicated to sort of figure out, where these things are. But that's basically the lateral forebrain bundle would be those fibers that are coming through the striatum that are going up to the cortex and punching through the striatum and going down into the spinal cord, but also down into the thalamus. So so there's the lateral forebrain bundle and the medial forebrain bundle. Okay, so that's pretty much the connections there, but we haven't talked about two other modalities. So the two other modalities are taste and smell. And so remember, like the visual system has the photoreceptors, and then there's a bipolar cell, and then there's a ganglion cell that goes in. And the auditory system, or somatosensory system, I guess we talked about. Somatosensory system has a little detector, which is actually the ganglion cell that goes in. And then the auditory system was yet different. That had a hair cell, and then that synapsed on a ganglion cell, like that. And then we get to the olfactory system. Oh. So the olfactory system is yet different. So you have these olfactory receptors, they're hair cells, and they project right into the central nervous system. And so where are they? So in a human, if you look at sort of a medial view of the cortex here, Jesus, can't draw a brain here. Let's draw that a little better than that. Okay, that's a better one. So if you look at where the olfactory bulb is, the olfactory bulb is sort of sticking out right here. There's a little bulb. And where is the olfactory epithelium? Well, here's the bone. It's quite thick, and then right next to the olfactory bulb, it gets very thin, and there's holes in it, like that. Then it gets thick again here, and that's called the cribriform plate. You probably don't write this down. Yeah, cribriform plate. That's where the olfactory epithelium axons punch through to get to the olfactory bulb. And so the olfactory epithelium is sort of sitting right here. This is like how you smell. So your nose is way down here. Your eye is sort of sitting here, and your nose is down here. There we go. So it's at the very top of your nose. And so those guys just grow through the cribriform plate into the olfactory bulb. So if you're getting your COVID test, they're not supposed to stick the swab straight up. There's been cases of where the testers have punctured the cribriform plate, causing cerebrospinal fluid to leak out. So they're supposed to aim for your ear when they do those deep swabs. It's a bad thing to go up like that. Those deep swabs are supposed to aim for your pharyngeal cavity toward your ear. So if somebody's like coming at you like that, say, no, I think my professor told me to sort of aim for the ear. So that's how the olfactory receptors get in there. They're constantly being renewed. And so, after a hard day of smelling all the crap in the world, some of them die off, and so they're regenerated, and then they grow back into the olfactory bulb. So that's how the olfactory system, and that accounts for most of your quote unquote taste. Almost all of your taste is the olfactory system. The taste buds is the last modality. So here's T. Those are actually cells that sort of sit in the tongue, and then they're more like the auditory system, where there's a ganglion cell that goes into the central nervous system. They're also renewed. If you eat a lot of hot food, you can mow them down, but that's okay, because the ganglion cell will say, just make a new one. And so they're also being renewed all the time. So it turns out that the olfactory system and the taste system strongly get into the limbic system. Not surprising, because you need to eat salt, and you need sugar has a lot of, your whole body runs on sugar, and so you need something that can detect sugar. So if you look at how those things get in, we got another. We're almost done. 1, 2, 3, 4, 5, 6. Just only 9 more things to go. So if we look, how does the let's do taste first. So here's some taste buds up here, and those project into something called the solitary nucleus, because if you look in a human brain, it looks like a little dot surrounded by a little gap of fibers. And then that projects to something called the parabrachial nucleus. So what are those? So these are the taste buds. Taste buds. and that goes to the solitary nucleus. And that is, if you were taking a neuroanatomy class, you'd see the solitary, if you look in the brainstem like this, you'll see this little dot there, the solitary nucleus with some fibers around it, and it's like a little tube going through the brainstem. If you look at something that has a really good taste system, like a goldfish, they can taste with their face, and they can taste with their tail fin. Their whole body is covered with taste buds. They're just swimming around, tasting stuff. Is that good stuff? Good stuff to eat? Those guys have like an incredible input structure for the taste system. It looks like a superior cloaca. It's actually bigger. It's stunning. It's bigger than their forebrain. Just the input structure for taste, and it's layered. It's got maps in it because it has maps of the whole body, like where all the taste buds are on the body. But our solitary nucleus is not nearly as good as that. Okay, so then it goes to the parabrachial nucleus. The brachium is that brac means arm, and so it's the arms of the fibers that are connecting up the cerebellum. Cerebellum is huge, and so there's these giant arms that sort of come out of the pontine nuclei and go into the cerebellum. And so this is the nucleus that's near the arms of fibers going into the cerebellum, the parabrachial nucleus. And so taste buds go into that solitary nucleus, that goes to parabrachial nucleus, and that goes into one of the substructures of the amygdala. And so the amygdala has some direct access to taste, like, is that a good taste, bad, poisonous taste? Bitter tastes are often poisonous. Okay, so that's taste. And then the smell input comes from the olfactory epithelium, but it turns out there's two separate olfactory epithelia. So the first one is the one that I drew down here, which is the main olfactory bulb. And so that one, we've got, we have that olfactory epithelium, and that's projecting to the main olfactory bulb. And then that projects to piriform cortex, which sort of means pear-shaped cortex. So here's the olfactory epithelium. And then this guy is MOB for main olf bulb. And then this is piriform cortex. So that's piriform cortex, part of the cortex, a simple looking part of the cortex. It has a little simpler layer structure than other parts of the cortex. And so that's how input gets in to the hippocampus. So parts of the hippocampus get a strong input. It's going back there too. So there's the main olfactory bulb, and there's the olfactory epithelium. And we know that smells are very much capable of sort of bringing up memories, and so it can get into the hippocampus and somehow access memories in the cortex in a very, very direct way. But we're almost done. There's another structure called the accessory olfactory bulb. And so 1, 2, 3, 4. Yeah, okay, so we'll put, that's good. So we just got the accessory olfactory bulb here, and that gets input from this other epithelium that you haven't heard of. So this one is this guy is the accessory olfactory bulb. And if you look at a rat it kind of looks like somebody just pasted two olfactory bulbs together, because the olfactory bulb has a very characteristic structure. It's got like one layer of very big cells, and then these little tufts of the dendrites of those big cells, of the principal cells in the olfactory bulb. So it's a layered structure, and it's sort of formed into a bulb. And then it looks like there's just another one just pasted in there. You can clearly see it's the same kind of structure, but it's just like sort of slammed in there. It's smaller. That's why it's accessory. And it gets input from something called the vomeronasal organ. Vomeronasal organ. And so where is that located? So the vomer is a bone in the roof of the mouth. And so there's actually a separate olfactory epithelium sort of mounted up there, sort of tucked inside the vomer. And that one is particularly involved. This is the olfactory organ that a dog is using when it's kind of following a scent. And this one only really works really close up. So it doesn't really work. It's not a distance olfactory. This one is the main olfactory epithelium is more like an actual sort of distance organ that can pick up odors kind of on the wind. This one is just mainly picking up very close odors. And it's in any animal, virtually all animals, like say, take an elephant. Now, if an elephant smells, like a male elephant smells the urine of a female elephant in estrus with his vomeronasal organ, he will make there's a particular expression, it's kind of like a raising his upper lip, even something like an elephant, to indicate that this pathway has been activated. So this is particularly involved in sexual smells, and that thing also gets in to the amygdala. So there's some question as to whether humans have a vomeronasal organ, and for a long time people said, no, we don't have a vomeronasal organ because we're not sort of like animals, and we've sort of bypassed all that. But it's probable that we do have a vomeronasal organ. Another way to remember the vomeronasal organ is snakes. You probably weren't expecting snakes. So if you've ever seen a snake, what do? They stick their tongue out and then they wave it around, and then they put it back in. But you didn't see what they were doing with the tongue when they put it back in. So they have a little forked tongue. They stick it out, they shake off odorants from it, and then they wave it around a little bit to pick up some odorants. They pull it back in their mouth, and they stick it up into their vomeronasal organ to sort of sample it. And then they do it again, shake off the odorants and do it again. So that's actually why snakes have that little funny tongue behavior. They're actually sampling odorants and sticking them into their vomeronasal organ to detect them. And it's very important to a snake. You lesion the accessory olfactory bulb or the vomeronasal organ in a snake, and a snake has no reason to live. It's kind of like, what's the point? It's like I can't smell. So it's a very important organ. And when we talked about the visual system and the auditory system and somatosensory system, we talked about maps. Turns out there's maps in the olfactory bulb and the accessory olfactory bulb. It's less studied there. Mainly it's been studied here. So there's actually maps of odors. Odor map. There's a specific odor map. So different parts of the olfactory bulb become active for different kinds of odors. There's no map here in the olfactory epithelium. So all the different odors are just kind of sprinkled around the olfactory epithelium. But when they go into the olfactory bulb, they sort themselves into different odors. And so there's actually a map in both olfactory bulbs. There's even a callosal, it doesn't go through the callosum, but there's a projection from one olfactory bulb to the other one that is a map-like projection. So even something like odors involves map-like processing. Okay, so that's the overall picture. So approximately, how does this work? So again, like I said, we don't really have a good picture of how this whole mess works because everything's connected up. But in general, say your hypothalamus detects I'm getting hungry. So it detects like blood sugar has gone down, or maybe I'm getting thirsty because the blood fluid balance isn't correct, because it turns out the hypothalamus has access. It has access to the blood. There's parts of the hypothalamus that are sort of permeable, and so there's something called the subventricular organ. It's like a little piece of the hypothalamus that actually samples the fluid in the cerebrospinal fluid, and it can sort of tell, what's going on with the body. So say the hypothalamus determines you're hungry, and so then you feel like I think I want to get Thai food, and so then how does that work? Well, the hypothalamus probably doesn't know where the Thai restaurant is located. So somehow, through some combination of all these connections, it's got to get back into the part of the cortex that actually knows where the Thai restaurant is. And that's probably a good chunk of that is probably in the neocortex. So how on earth does that work? Or like, who's running what? Not clear, but it's very clear that things that have been detected about the state of the body by the limbic system somehow can percolate back into the sensory, higher level sensory structures, motor structures, in order to actually access information that's not in the limbic system. So the limbic system doesn't know what things look like. It doesn't have fine, detailed structure of shapes. It doesn't know what corner to turn on when you come to a particular street. That's almost certainly stored somewhere up here. So, and you could argue maybe partly in the hippocampus, but how that sort of whole process works, not entirely clear, but we know it does. So things that are detected about internal state here can percolate into the rest of the system and access information all over the place in order to get things done. And just think about, like, an animal gets thirsty, it has to know where the water hole was, or maybe it has to remember, if I just wait until morning, I can sort of lick a few of those water droplets off the dewy leaves. Even in a desert environment, there's going to be dew in the morning. It has to remember, like, where are some big leaves where they can get a lot of water from. So those kind of things are not directly in here, but we have to access them somehow. So the limbic system it's not a completely encapsulated thing. It's hooked up to, it gets its own sensory input. It can access a lot of motor structures. It can sort of talk to the whole rest of the cortex. It talks to different parts of the thalamus. So it's just a sort of a convenient way of letting Marty make one diagram that fills the whole board. Okay, so any questions? We'll just do 3 minutes on the pedunculopontine nucleus, but any questions about the limbic system? All good on the limbic system. This covers the main parts. I will not ask you to reproduce this on a test, but I just want to sort of lay out all the main actors that we know are involved in all these things. And you can see there's a lot of connections between different parts of it. So, stunned silence as usual. That's all okay. The limbic system, eating and drinking. It's the 4 F's: fighting, feeding, fleeing, and mating. All good. Okay, so last but not least. So for many years, I would tell people, there's the limbic midbrain, it's got the pedunculopontine nucleus in it. But I didn't really know where the pedunculopontine nucleus was exactly. So so a couple years ago I said I should just upgrade my lectures and just like learn something about the pedunculopontine nucleus. And so I went to the pedunculopontine literature. And now the funny thing is if you go to read about something, you know, it will always be the center of the universe. And so here's the pedunculopontine nucleus. So it's a big nucleus. So where is this guy? So this guy is. This is, you know, connections of PPN, pedunculopontine nucleus. So where it's located, if you look in the brainstem, you come down the brainstem, and then there's a big bulge of the pontine nuclei like that, and this thing is the peduncle. And what's the peduncle? Cerebral peduncle. It's basically just a giant bundle of fibers going down into the spinal cord, like all the motor cortex, red nucleus, reticular formation. You know, lots of stuff are going to get into that thing and go down to talk to the spinal cord. And so that's the pedunculo part of the pedunculopontine nucleus. And so the cerebellum is over here. So this is all, you know, this giant human cerebellum sort of sticking on top. And so where is the pedunculopontine nucleus? It's kind of located like right about there, and pretty close to the. So that's the pedunculopontine nucleus. So it's near the top of the pontine nuclei, just under where the peduncles start to split up as they go through the pontine nuclei. So as they go through the pontine nuclei, the peduncle kind of splits up into a bunch of things, and then it comes back together down below. So that's the pedunculopontine. So it's the center of the universe. And so what's connected up to it? Well, it gets a bunch of cortical input. Why not? I didn't realize that. So cortex, a whole bunch of cortical areas project down to it. And it tends to focus on somatosensory cortical areas, but it gets some visual input and somatosensory and auditory input as well. So that's the cortex. And then it has a whole bunch of projections to motor structures. So it itself doesn't have any strong projection down to the spinal cord, but it goes to the vestibular nuclei, and it goes to the deep cerebellar nuclei, and it goes to the reticular formation. So the reticular formation, that was one of the major motor output pathways. And the deep cerebellar nuclei, like the dentate nucleus, those guys, now we pitched them as something that the cerebellum projects to, but it turns out the deep cerebellar nuclei are like a little motor cortex because they project to the red nucleus. And so here's the red nucleus. And the red nucleus is like the original motor cortex. And so the deep cerebellar nuclei can get input from the cerebellum, but they can also get input from the pedunculopontine nucleus. And so then that can go to the red nucleus, and then once it goes to the red nucleus, it can go to the spinal cord and do stuff. So here's, you know, spinal cord. So there's a projection, big projection from the red, crossed projection from the red nucleus to the spinal cord. And there's a big projection from the reticular formation to the spinal cord. And it goes to the vestibular nuclei. So as we said, the vestibular nuclei, they're a lot more than vestibular. because they're getting input from the limbic system, pedunculopontine nucleus. So like the hypothalamus, I mean, that's the thing about the brain connections. There's not that many parts. 400 is not that many parts, but they're all connected up. Like the hypothalamus can talk to the pedunculopontine nucleus, and then that can actually go to the vestibular nuclei, which can then, go out to the spinal cord. But it turns out being the center of the universe, the pedunculopontine nucleus is also connected up to the globus pallidus. So how did that happen? So it's actually connected up to both chunks of the globus pallidus. So here's both chunks of the substantia nigra. So this is the substantia nigra, pars compacta, that's the dopamine part. But it's also connected up to the substantia nigra reticulata, that's the GABA part, the inhibitory gating part. And so obviously it could thereby get into the the caudate putamen. So let's put CP over here. And then eventually get to motor cortex. So there's projections, basically back and forth with both of these guys. So it can get involved in sort of gating actions, deciding, what to do. How hungry are you? Are you hungry enough to sort of overcome like a dangerous situation and sort of do something anyway? With complicated decisions that have to be made. It also does go to the globus pallidus. So it only goes to the internal segment. So here's the globus pallidus internal segment, but that's another gating structure. The GABA one down here, the substantia nigra reticulata, was the gating one, but the globus pallidus internal segment was the gating one. And so this thing projects to the globus pallidus, and then that can project up to the ventrolateral that's the motor cortex one, and then this is the premotor cortex part of the thalamus. Remember, it's inhibiting them, and that can get up to the cortex, where it can obviously go down all the way to the spinal cord. And last but not least, being the center of the universe, it's also connected up to the subthalamic nucleus. Remember, that was the one that, STN, that was the one that inhibits funny movements of your arm. That's the function of the subthalamic, just joking, this function of the subthalamic nucleus. If you lesion the subthalamic nucleus, what happens is there's not enough excitation of the globus pallidus internal segment, and so then that results in too many movements getting through, and so you get all these funny movements. So the pedunculopontine nucleus is talking to that. So you can clearly see the pedunculopontine nucleus is the center of the universe. But the interesting thing here is just to sort of see how it sort of breaks your normal boundaries of how you think things are hooked up. Like the red nucleus, no one ever expects that the pedunculopontine nucleus is talking to the red nucleus. But this shows that there's a substantial parallel pathway for sort of affecting movement that's coming directly, out of these limbic structures, in addition to all the direct outputs that are coming from the cortex. Okay, you definitely do not have to remember the connections of the pedunculopontine nucleus. I can't remember them myself, but it's a nice big hunking nucleus there. Like I said, when I was teaching neuroanatomy, I'd see it and I'd go like, yeah, there it is. Big thing, have no idea what it does. But people have started studying it more recently. Okay, so this is kind of a heavy-duty neuroanatomy lecture without any of the structures, but just sort of the the basic connection. And the important thing is to sort of keep in mind, like, what is the main point? Is we have all these devices here that are in the, especially in the hippocampus and in the hypothalamus, like the mammillary bodies, monitoring where your head is. How hungry am I? How angry am I? How thirsty am I? What direction is my head pointing in? Where am I relative to the other things, the main features of the environment? Where exactly am I located? And all those kinds of internal state monitoring functions somehow have to sort of like tell the other systems, how to fix up your internal state, how to find food, how to find water, how to avoid different threats, how to navigate around in an uncertain environment. And again, I've said this hundreds of times, but people go in one place. They study. If you read the pedunculopontine nucleus papers, they'll just look at what happens in the pedunculopontine nucleus. But the problem is it's hooked up to the whole world, and so how does the signals coming to the deep cerebellum nucleus that come out through motor cortex interact with signals coming into the deep cerebellum nucleus from the cerebellum, or from the cortex going through the pontine nuclei to the cerebellum? Well, they typically won't have studied that, but those are all critical for how the whole thing works together. Okay, so any questions about the limbic system overview? All good. Good. Pretty good. Yeah, I mean it's good. The thing is, there's way too many words that we've gone over this semester. You can't possibly remember them, but it's good to just sort of see what's, just get an overview of what's there, and then you don't have to learn the whole thing. And typically, if you're gonna go and study something, if you're going to study part of the hippocampus, you're going to focus on the hippocampus. There's no problem with that. That's how we figure out stuff. But it's also good to sort of like see the overall picture, keep the overall picture in the back of your mind. You don't have to like remember it all. Okay, Stan, I think we'll end the recording there.